Going to talk about nucleic acids in a cell tonight. First, let's talk about order from disorder. The conversion of complex molecules, the transfer of heat, and the low efficiency of the whole process. Some of the disorder we experience is the conversion of complex molecules like glucose into energy. This has a very low efficiency. So a huge part of the energy from the fuel sources that we transform is transformed into heat, most of which dissipates into our environment. The transfer of heat increases the entropy, as well as converting complex biomolecules into smaller molecules. So the high degree of organization of living beings is supported by the constant input of energy and is offset by an increase in the entropy by the being's surroundings. So we have our complex molecules. We have kinetic energy, we have carbon dioxide, we have water, we have heat, we have everything we need there. An activity that we have here for cellular respiration, it's going to be an activity where they're going to see that we're going to take in and use disorder and make order from it, making our cells do what they're supposed to do. The task here is for them to find something cells need to break down the sugar so that we can use the energy from our sugar molecules. So first they're going to start with plants. We're going to give them empty frames and they have to see what's missing. Leave the door of the stomata open and the oxygen atoms from earlier outside. So the students will find that they need oxygen in order to complete the molecules and should figure out that it's from the air outside the leaf and it has to enter. And then the carbon dioxide and the water are going to be released through the stomata. The second stage of this is going to be when we talk about animals. They're going to reassemble the sugar molecules for this round, put all the materials back in their starting places. They're going to explain that the animal cells need energy, and to get this, by breaking apart sugar molecules. But animal cells cannot make their own sugars the way that plant cells can. So where do animal cells get their sugars? That's a discussion. They get it by eating their plants. So we tell the students that the leaf they are a part of is about to be swallowed by a hungry herbivore. The leaf is getting chewed up and digested. Then the sugar molecules that were contained within that leaf are passed to the cells in the body. So we take down the stomata sign and the stem sign. We tell students that the classroom now represents the animal's body. Each table is a cell within the animal. The cells need to break apart the sugars to release energy so the animal can use it to keep moving around. Just like in plants, the processes will release carbon dioxide and water. What are they missing to make this happen? We know it's oxygen. Where will the animal get that oxygen? We know it's by breathing it in. So put a new sign over the door that says lungs. Now go through the respiration process again. This will be the very same process as it was for plants. The only difference is the oxygen enters through the lungs instead of the stomata. Students should bring oxygen in through the lungs and release the carbon dioxide and water produced in the process of exhalation from the lungs. So it's a good exercise for them to understand how that all works. Energy coupling is the pairing of two biological reactions, which means the energy created from one reaction is used to drive the other reaction. As seen in the slide, ATP hydrolysis is coupled with polymer synthesis as an example. Exergonic reactions are often paired with endergonic reactions. Exergonic reactions are actually energy reactions that give off energy, and endergonic reactions are ones that take in energy. Because the energy released from the exergonic reactions can be channeled down to the endergonic reactions, that's how the endergonic reactions can be driven by the exergonic reactions. So if you look at the ATP and the ADP and use of energy, it shows how that one can drive the other. Nucleic acids play an important role in all cells and viruses. We're going to talk about where they are and how they function in our cells. An important function of nucleic acids is the expression and storage of genomic information. Our deoxyribonucleic acid, otherwise known as DNA, carries information the cells need to make proteins. There's another important nucleic acid, ribonucleic acid. RNA comes in many forms and has multiple roles in the cell, including protein synthesis. So if we're looking at our slide here, we have DNA, double helix. It's got 4 base pairs. It's got its deoxyribose sugar. Here are its base pairs. It's got a phosphate group. We look at ribonucleic acid. It's got a single strand. It's got its sugar phosphate backbone. It's got its ribose sugar versus the deoxyribose sugar over here. It's got its 4 base pairs. Now we have uracil versus thymine here. So they're not exactly the same. Down here, if we click on this URL, there's actually a game that can be played to help you run through the process of protein synthesis. That will help would help lead you through the process of protein synthesis. How does this work? How does the DNA get taken apart by the RNA? The mRNA reads the DNA, which gets taken to the tRNA with the ribosomes, and then they start reading it, and then they create new chains of DNA, and they put them all together, and then they get put into different cells, and then we create our new cells so that every cell has the same DNA. This particular link has a game that'll lead you through that. Carbohydrates. The definition, classification, and structure and functions. There's monosaccharides, disaccharides, and polysaccharides. Carbohydrates include simple sugars as well as polysaccharides. Okay, so we're talking polysaccharides. Your simple sugars are going to be your monosaccharides. That would be your glucose, your fructose, your galactose, things like that. Your disaccharides are going to be your sucrose and your lactose. So glucose is one of the simple sugars that is a major nutrient for the cell. Polysaccharides are the storage form of the sugars and they help form structural components of the cell. They, along with shorter polymers, actually act as markers for cell recognition processes. So we look at the polysaccharides down here, and they're going to include cellulose and starch and glycogens. So those are going to act as markers for cell recognition processes. Your disaccharides don't have a big role like that, but then your glucose is going to act as one of the major components for energy for the cells. And the fructose, things like that, they're going to get broken down. They're going to be used as energy for the cells.